Hello dear students, I'm Dr. Moin. In this video, we are going to discuss about analytical problems and their solutions. Means in this video, we'll see where from the analytical problems arise and how do we find their solutions. Before starting the video, if you didn't subscribe my channel yet, then subscribe it right now. And also press the bell icon so you may get in touch with my upcoming videos. And here are the contents of current video. First of all, we'll see what is analytical chemistry. That is, we'll see what is done in analytical chemistry. Then we'll see the analytical problems, what are analytical problems and how their solutions are found. Then we'll see analytical process, means what steps take part during a whole analytical process. Then we'll see the nature of analytical methods. Definitely there are different types of analytical methods. So what types of those methods are. And we'll also see in the end the classification of these analytical methods. So let's get started. So let's start with analytical chemistry. Analytical chemistry is concerned with the chemical characterization of matter. We know that anything which has mass and occupies space is called matter. So we might be interested to know the chemical composition of matter or substances present around us. And chemical composition means that substance or matter is composed of what type of elements or compounds. So analytical chemistry is all about the chemical characterization of substances around us. And it answers two important questions. First one is, what is it? Mean that thing is composed of what type of elements or compounds, and this is called qualitative analysis. And second thing is, how much is it? Mean that element or compound is present in what quantity, and this is called quantitative analysis. So both of these, qualitative analysis and quantitative analysis, make a complete analysis. We are surrounded by chemicals around us, and knowledge of chemical composition of many substances is important for us in our daily lives. So we are surrounded by chemicals all over around us, and we might be interested to know what are the chemical composition of those compounds or those substances. So we must have to opt the analysis or analytical chemistry. It plays an important role in nearly all aspects of chemistry. For example, in agricultural fields, like we might be interested to know what is the composition of soil so that we can apply some suitable fertilizer. In the clinical field, for the analysis of blood or urine sample. In the environmental field, for the analysis of air, water or soil, etc. In forensic chemistry, forensic sciences, for the identification of crimes or criminals. Then in manufacturing field, some industry is manufacturing what substance, so we might be interested to check whether the composition is right or not. Then in metallurgical field, and finally in the pharmaceutical chemistry. So all these fields require the analysis. So analytical chemistry plays a vital role in all these fields. And here are some more examples from different fields. For example, the nitrogen content of a fertilizer determines its value. means different nitrogenous fertilizers, they have different nitrogenous level. So how much nitrogen is required in our soil, so that is determined totally by the analysis of fertilizer and that of soil. Then food, it is from food industry. Foods must be analyzed for contaminants, like there could be the pesticide residue, so we can analyze the food, that how much pesticide residues are inside, whether they are toxic, up to toxic level or below that. And for essential nutrients, and we may also analyze foods to test their essential nutrients like vitamin contents and other things. The air we breathe must be analyzed for toxic gases like carbon monoxide and other gases. Blood glucose must be monitored in diabetics, and in fact most diseases are diagnosed by chemical analysis. Then the quality of manufactured products often depends on proper chemical properties, and measurement of the constituents is a necessary part of quality assurance. So to maintain the quality of the product, definitely we must have to perform analysis on the finally prepared products. The carbon content of steel will influence its quality, so it's from metallurgical field, that the quality of steel can be determined from that how much carbon content is present inside. And last one is from pharmaceutical side, the purity of drugs will influence their efficacy. I mean, how much that is pure, so definitely that will be more efficient, or vice versa. So concluding this, we can say that analytical chemistry provides the methods and tools needed for insight into our material world. So we can look around us into different things, that what they are, and through what things they are composed of. So analytical chemistry, actually it provides the methods and tools to do all this process. For answering 4 basic questions, and why we want to do all these things, to answer actually 4 basic questions about a material sample. And what are they? What is it? What the thing is, and where from it? Then how much, mean the things are present in how much quantity, it's related to quantitative analysis. And also what arrangement, structure or form. So analytical chemistry, there are some techniques which can also help us to know that what is the structure of elements or compounds present inside that matter or substance. So these 4 questions could be answered by using the methods and tools provided by analytical chemistry. Then analytical problems and their solutions. Many problems in analytical chemistry begin with the need to identify what is present in the sample. That is qualitative analysis. A lot of problems are of us which arise in our daily life, they are related to the qualitative analysis, means what is present inside the sample. For example, identifying the product of a chemical reaction. We have performed some chemical reaction, and definitely we are interested to know that what has been formed there. So it's a qualitative analysis. Another example is that there could be the screening of an athlete's urine for the presence of some performance enhancing drug. So means there are going some games, and we are interested to know that whether the athletes have not taken some performance enhancing drugs. So there could be the screening of their urine, so it requires some qualitative analysis, etc. Previously, these tasks were being performed by the use of some simple tests, but these days modern techniques like infrared spectroscopy and nuclear magnetic resonance NMR spectroscopy are also in practice. So by the use of some conventional methods and the modern techniques, we are performing all these qualitative analysis these days. While some other problems may involve quantitative analysis as well. For example, measuring the concentration of glucose in the blood, or measuring the concentration of chromium in steel. So these are the examples of quantitative analysis. While another area, and that is characterization analysis, and it deals with the development of new methods for characterizing physical and chemical properties, equilibrium constants, particle size, and surface structures. So the purpose of a qualitative, quantitative, or characterization analysis is to solve a problem associated with a particular sample. Means, in chemistry, we can solve a problem by performing these qualitative, quantitative, and characterizational analysis. Now we will see an analytical process. So there involve various steps in an analytical process. Analytical process means the process to solve a problem. So actually there involve a large number of steps in an analytical process. And the analytical chemist should be involved in every step. So analytical chemist is required in all the steps. These steps are being discussed here. So now we will discuss all of these steps which make this whole analytical process. And here is the first step of an analytical process, and that is define the problem. And here are different factors. So what is the problem? So what is the problem? And what need to be found mean what we want to extract through the solution, whether we want to get some qualitative or quantitative information. Then what will the information be used for? So for what purpose the information is going to be used, and who is going to use this information? Then when will it be needed? At what stage the information is required? Then how accurate and precise does it have to be? The information should be how much it could be the accurate or precise. Then another thing which is very much important, and that is what is budget, because within the limit of the budget we have to solve this problem up to that precision and accuracy level. Then the analyst should consult with the client to plan a useful and efficient analysis, and this should also include that how to obtain a useful sample upon which the process can be applied to get the results. And here is the second step of an analytical process, and that is select a method. So on what factors a method is selected. So first one is what is the size of sample. So it also gives us direction for the selection of method. Then whether sample preparation is needed or not. So this also gives us some help us for the selection of some method. Then what is the concentration and range? So in what concentration the sample is required. So this also helps us to select a method. Then accuracy and precision needed. How much our results should be precise or accurate. This also leads for the selection of method. Then what tools, instruments are available to us. So on this base we can select a method. Then what instruments we are selecting. So whether we have expertise to use that one. So This also gives us insight for the selection of that method. What will be the cost of the process? So it's all related to the budget. Then speed of method. So different instruments, they have different speed for the analysis. So this also gives us for the helps us for the selection of a method. And are methods available in the chemical literature? Mean we have selected an instrument, so whether the protocol is available in the literature, mean how to run our instrument, how much sample should be taken, what temperature programming or what other things could be set on the instrument. So whether all these things are available in literature or not, or we have to design the whole protocol by ourselves. So these are the things which help us for selection of a method. Then third step is obtain a representative sample, and this may include the sample type, mean what is the physical state of the sample, whether it's solid, liquid or gas. Then we can see its homogeneity, mean whether it's homogeneous or not, and then we can see the size of the particles of sample. Then step number 4 is to prepare the sample for analysis. Again, we will have to see whether the sample is in solid, liquid or gaseous state, whether we need to dissolve it in some suitable solvent or not. So it all depends upon the technique, the instrument, whether we need to convert the sample into ash, or we need to do digestion for the sample. Then whether we need some chemical separation or masking of interferences needed. There are sometimes present some interfering bodies, so which can disturb our results. So we need to add some masking agent, so that can mask an interfering body, so we can get the quite accurate results of our sample, of our analyte of interest. Then whether we need to concentrate our analyte or not, whether we need to adjust the solution conditions, which may include the adjustment of pH or addition of some reagents. Then whether we need to change the analyte for detection. This actually, some of the analyte cannot be analyzed by using of some instrument as such, so we need to derivatize those substances in which we change their nature, and in this way the derivative can be analyzed by the use of some technique. So this is all about the preparation of sample for analysis. Then step number 5 is to perform any necessary chemical separations. So sometimes there could be required some chemical separations, and there are different methods which can be opted to perform these chemical separations. And here is the list of these methods, which depend upon the analyte and the conditions actually. So these methods are distillation, precipitation, solvent extraction, solid phase extraction, chromatography and electrophoresis. So by the use of any of these methods, we can perform any necessary chemical separations if required. Then step number 6 is to perform the measurement. So it requires different steps. So if we are using some instruments, definitely whether it's the instrument is working properly or not, we need to calibrate our instrument. And to run the process, we need different controls, standards, we need blanks if we are using spectrophotometers or chromatographs, etc. And then we run our sample in replicates, generally in 3 replicates. The sample is run through the instrument so that our results could be reliable. So these are different steps which are taken during measurement. The last step is to calculate the result and report. So we do various statistical analysis in which we perform different tests on it to check the reliability of our results. Then we do report the results with informations like what are the limitations or accuracy present in those results. Then we do interpret carefully for our intended audience, in which then further the results are critically evaluated, and finally we reach to the point that whether iterations are needed, means whether we need to repeat the whole process or whether it's okay. So this is all about the analytical process. And lastly, we are going to see the nature of analytical methods. So depending on the nature of analytical method, it can be classified into qualitative and quantitative method. So the analytical method, whether it's performing for us the qualitative analysis or quantitative analysis, so it may be classified as qualitative and quantitative method. However, analytical methods can also be classified as classical and instrumental methods. Let's see this classification. So first of all, classical methods. So in classical methods, how do we perform the qualitative analysis? So it is done by identifying the color, odor, or by the use of indicators, or by measuring the melting points, boiling points, etc. And how do quantitative analysis is performed in these classical methods? So it may be done by calculating the mass or volume, which makes the basis of gravimetric analysis, volumetric analysis, etc. So this is all about the classical methods. And then instrumental methods. So how qualitative analysis is being performed in instrumental methods? So it's being done by the use of technique like chromatography, electrophoresis, and by identification by measuring physical property, like it's being done in spectroscopy, electrode potential, etc. And how quantitative analysis is being performed in these methods? So it's being done by measuring property and determining its relationship to concentration, like it's all being done in spectrophotometry, mass spectrometry, etc. And most often, same instrumental method is used for both qualitative and quantitative analysis. Mean a single instrument is giving us both type of information, that is qualitative and quantitative information. And then a general classification of important analytical methods. So here is a table in which there are different methods and what property is being measured into them. So first of all is gravimetric methods. So what is done there? So weight of a pure analyte or of stoichiometric compound containing it is being measured. Then in volumetric method, volume of standard reagent solution reacting with the analyte is measured. Then in spectrometric method, intensity of electromagnetic radiations emitted or absorbed by the analyte is being measured. In electrochemical methods, electrical properties of analyte solutions are being measured. And in chromatographic methods, physicochemical properties of individual analytes after separation are being measured. And in the end, there's the types of instrumental methods. So here's the table, so in which different properties and they are being measured in what type of techniques. So first of all is radiation emission, and it is being observed in emission spectroscopy, fluorescence spectroscopy, and phosphorescence spectroscopy. Then radiation absorption, it's being measured in absorption spectroscopy, spectrophotometry, nuclear magnetic resonance spectroscopy, and electron spin resonance spectroscopy. Then radiation scattering phenomena, it's being observed to calculate the turbidity and in Raman spectroscopy. Then radiation refraction, so refractometry and interferometry, so it gives us information like this. Then radiation diffraction, and it's being observed in X-ray spectroscopy. Radiation rotation, it's being observed in polarimetry. Then electrical potential, and it's being observed in potentiometry. Electrical charge is being observed in coulometry. Electrical current is being observed in voltammetry, amperometry, polarography. Electrical resistance is being measured in conductometry. Then mass is being calculated in techniques gravimetry. Mass to charge ratio is being calculated in mass spectrometry. Then thermal properties are being observed in thermal gravimetry, calorimetry. Then rate of reaction are being observed in flow injection analysis. And then radioactivity, so these are being observed in the method like isotope dilution. So dear students, this was all about current video, but still there are a large number of videos in the pipeline. So to get them in touch with all of those videos, you need to subscribe my channel. So thanks for watching. Thank you very much.